Hello, everyone. Welcome to today's session. We will be starting this program shortly. Thank you for joining us today. It's great to have you all here. Hello, everyone. So we will be starting in about 15 more minutes. Thank you for joining today and for your patience. We will record the session again and post it in our YouTube channel if you're interested to rewatch today's session. And during the session, please keep your microphones muted and your video turned off. If you have any questions, the chat box will be open and our team members will be answering any questions that you have. So thank you again. Hello, everyone. It's great to see everybody here. So, we will be starting in just about a few more minutes. Thank you for joining us again. Welcome to this webinar where we will speak about our new product that we have been working on, the OmicsLogic Code Playground. Code Playground is the place to learn and practice bioinformatics coding by following interactive tutorials and completing challenges in R and Python. These courses and exercises will help you understand how to analyze and visualize omics data to extract meaningful insights from large biological datasets. We hope that you too will use this resource to learn, practice, and achieve bioinformatics greatness with concise exercises and interesting challenges right in the comfort of your browser. My name is Deepsha Biswas, and I am the OmicsLogic Community Manager responsible for daily interaction with thousands of users that are part of our growing community. Before we begin, let me introduce what OmicsLogic is and how the training resources we will discuss today have been developed and are used in the OmicsLogic training programs and for research support in academic institutions around the world. I'm honored to have been working with a team that collectively have amazing experience in this area, including the other co-founders of the company, Dr. Alfred Tauber, who comes from a medical background with expertise in oncology, immunology, and biochemistry of inflammation, has a long-standing recognition for his generous support of many international projects and research activities. One of these projects is the establishment of the Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. He has numerous publications on bioinformatics and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. These have been widely used in basic and translational research in biotechnology, pharmaceutical research and development, agriculture, and basic research. What we will discuss today is a result of hard work by some of the people mentioned here. Julia Panov is an expert bioinformatician and helped prepare many of the materials we will use. Avi Titievsky is a computer scientist and leads the development of the T-Bioinfo platform that we use in our OmicsLogic programs. Dr. Mohit Mazumdar is in charge of business development and a project mentor. Joshua maintains the educational portal. And of course, there are many more that I simply do not have the time to mention today. OmicsLogic programs are a result of close collaboration between Pine Biotech, an innovative big data analytics company based in the US, and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. As a result, the OmicsLogic programs have been thoroughly tested around the world and are known to lead to excellent outcomes for students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in data science and biomedical research. Now, I would want to invite Ilya Brodsky, the CEO and co-founder at Pine Biotech, to talk more about this platform with you. Over to you, Ilya.
Thank you very much, Pipsa. And I'm happy to see many of you that we've gotten to know in the past programs and in different types of situations. So what I'll do today is briefly introduce you to this new and exciting project for us, which was the development of the Code OmicsLogic Playground. So as Pipsa mentioned, my name is Ilya Brodsky. For those of you that are joining for the first time, I'm the co-founder and CEO of Pine Biotech. And I want to tell you a little bit about what Pine Biotech is seeking to achieve. Our vision is to enhance human health and well-being by enabling biological discovery with relevant data, solutions, and expert support. And so we've been working for the past couple of years on developing a number of resources that will allow biologists to easily manage, process, analyze, and visualize their data. So as a result, as you know, we have developed a number of educational resources that include our T-Bioinfo platform that was designed and developed at the Tauber Bioinformatics Research Center. And our team has been working on applying some of the methods in that platform to allow a seamless transition from learning about bioinformatics to specializing in different bioinformatics domains and applications, to then translating that knowledge and those skills to independent projects that could be supported by our team. And so as we looked at the field that we're in, we realized that we discovered that these tools are basically creating a solution for the gap in research and discovery. And an important part of that solution is coding. So we realized that as students become proficient in the logic of bioinformatics and are able to apply different methods for processing big data, they need to be more empowered with downstream analysis tools that include a typical collection of different libraries and code blocks in R and Python. And so then we started working on expanding that idea into a product, into a solution that I think fits in very nicely with the rest of the tools that we have. So as a result, working with our team, we've designed and developed this OmicsLogic Code Playground. The playground allows a user to learn about different types of challenges, understand the code approach, and then actually practice that code within the browser. And so the way that we kind of thought about this is that we want to allow people to understand what are some of the challenges, learn the different pieces of code and the syntax, to be able to start off with a template that they can modify, including the different outputs and the settings, and then learn to apply that to a data set of their own. And by incorporating this into the other solutions that we already have, into the T-Bioinfo platform and into the educational portal, we are able to really fill in that gap that allows biologists and people with a minimal technical background to really develop coding skills as they work on projects of their own. And so as we have done with the T-Bioinfo platform, our objective over the course of the next several months is to really supplement the areas of analysis for genomic, transcriptomic, epigenomic, metagenomic, and proteomic data with these tools and methods that we will provide tutorials on in different languages like R and Python. So today, I wanted to start off with a brief demonstration of how it actually works. So for all of those that are new to the OmicsLogic playground, I want to ask you to go to codeomicslogic.com. And Vipse, if you could please paste that in the chat. So if you could go to this website, the registration is free. So if you have not registered already, you can just go ahead and register for free. And you will see that in each category of courses, we have some free resources that you can leverage right away. So please let me know once you've been able to reach this page, and I'll continue from there. Once you create your account and you log in, you will have access to different courses. And so we wanted to start with this set of courses that allows anyone to get started with coding in R, Python, an introduction to data science, and then several examples applied to genomic data, metagenomic data, and transcriptomic data. Our goal is to add new lessons and courses every week to this platform. So we hope that this is just a resource that we're getting started with. And today I will also show you how easy it is to add new lessons and tutorials. So for those of you that are interested in working with us on expanding this resource, we are happy to connect with you and discuss what are the opportunities to actually expand this resource and utilize in different domains. So right now, we are applying this to the domain that we're in the OmicsLogic training, and I'll talk a little bit more about how we do this in the OmicsLogic programs. And if you have an idea for a tutorial in R or Python, I will show you how easy it is to add a new resource onto this platform and make it available online. So as you join, I want to demonstrate one of the examples. It's not going to take a very long time, but we can actually try to do this together so that you really get a sense of what can be done in a very short period of time and how the courses expand your understanding of bioinformatics in application to different types of data sets and an in-depth look at the different types of challenges that are solved with these analyses. So let's go to Introduction to Data Science in R. You can see here a description of the overall course. This Introduction to Data Science in R is a part of our Data Science OmicsLogic training program, but it can also be standalone. Should we use this to create an account for Code Playground? Yes. Again, for those of you that already have an account on the educational portal, you can actually use the same email address, and I will show you how the two are connected. So they are interconnected. As you know, we use this point system to track activity, and all of those are integrated. And you will notice that within each course, there are going to be several lessons that are free and several lessons that are called premium. Now, this premium option comes at a minimal price. The subscription is $10 a month. And as I mentioned, these types of tutorials will expand as we move forward. Every week, our plan is to add new material to this site. So once you're in this course, let's open up dimensionality reduction, principal component analysis, and visualization. Just so that I have a sense of how many of you are with me, and adapt my pace accordingly. Could you let me know if you've reached this course so I know how many of you are following along? All right, excellent. Thank you very much. Okay, so principal component analysis allows us to study continuous data, for example, gene expression data, to understand how the gene expression characterizes samples, placing them into a single multidimensional space and reducing the dimensionality so that we can understand differences between samples or groups of samples in a scatter plot, in a visual output. So here we describe the input data. You can see the input data is a set of samples characterized by 15 genes with their gene expression values listed right here in the cells. A little bit about this data set. This data set comes from a cancer study that is focused on identifying different subtypes of cancer. So let's take a look at how we can apply principal component analysis in R so that we understand the syntax, the analysis approach, and what are some of the different options that you can modify and use to improve the basic visualization. So as you can read right here, the first thing that we have to do is load some libraries. As anyone who's used R before, you know that one of the major challenges is setting up your environment. And so that's the first thing that we want to eliminate by offering this platform. Instead of dealing with everyone installing the different packages and resolving dependencies and maybe not having the right version to be able to run something, we want to just jump into the code and understand what it does. So as you read through this, we will refer to the specific libraries, and then we will explain how the data is loaded in and transformed so that it is ready for the analysis. Now, what's amazing about R and many of the packages in Python is that the actual analysis could be done in a couple of lines of code. So it's really easy to learn and get started without being overwhelmed with how to actually structure your analysis. So what we do here is use a basic analysis with principal component, which is achieved by doing prcomp to the transpose data table that we loaded in. And a very simple output is just creating a scatter plot of principal components that looks already somewhat informative. Now, as we go through the rest of the code, you will see how we can improve the output, adding colors, adding the loadings for the different genes that we have or the features, and eventually plotting this in 3D. So let's take a look at how to do this. Importantly, we don't want to just provide you with an opportunity to rewrite the code, copy and paste, and then see the result. We also want to understand what this analysis does along the way. So let's take a look. Here, you have an editable piece of code that you can try running right away. So if you click on run, the code will run in the cloud and produce an output. Okay, so you can see that the output is being produced. Those images are going to load. And let's just take a look at what we did here. So we have a file. This file is stored on GitHub. And then we can read the table, convert it to a matrix, transpose the matrix, and then run our principal component. After we run the principal component, you see that we can plot it, or we can even print out what is inside of this result. Because R is an object-oriented language, we can actually print it. So we can add here print PCA. So let's run it again. And we see what is in the object. So in the object, we have all of our genes, and we have some principal component analysis results. So you can see here PC 1, 2, 3, etc. So what we do is we actually take this analysis and we can visualize it using the standard scatter plot option, which is shown right here, plot PCA X and Y, or we can use the ggfortify package, which allows us to create a ggplot object that is easy to work with to improve the visualization and add all kinds of additional information onto this single plot. So what's important here is that we've explained the code and explained the problem, and then we also demonstrate how you actually run it right within the browser, being able to modify different aspects of this code to be able to improve the visualization or even change the file that you're working with. So change the data set very easily. So here we explain some additional information about this code, including an insight into how to add some more information. For example, to output a textual result, which will be a file. So if you look at this new area, you can try running this code and adding this part of the script right here. If I run this as is, so if a student wants to just run whatever and complete all of the steps without thinking about the problem, they won't be able to. So you can see that I'm getting an error, and I can see what the error is. And the error, as obviously seen right here, is that the code is incomplete. And I even have the red parentheses here that show me that it's incomplete. So what do I have to do? Now the student or the participant who's trying to learn R really needs to go back and see, what am I trying to do and how can I do it on my own? So actually this part is very new. So this is going to be very simple to solve. Let's solve it together. So here we see what the code does. We load the library, we have the input file already, we define the data, we even have the sample names already. And PCA is also already mentioned here. But what we have here is printing the length of PCA result object. So how do we print the length of this object? We simply take this and place it inside the parentheses. So if I want to print the full object, I just do PCA result. If I want to know the length of this object, I have to put it within the parentheses. And here it says, add here the script to print the results table. Now, here I have how this table would be printed. So I can, for now, just copy and paste it right here. Let's clear the console and run it again. Okay, and here we discovered there's another problem. So let's see what the issue is. Okay, right here. So make the plot from the previous step. So we also need to make the plot. So how did we make the plot previously? We did autoplot for the PCA, right? So we can just copy that right here. Let's place it here. Again, let's clear the console. Excellent. So our code is successful and the answer is correct. So for each time that we run this code, you're actually rewarded with points, encouraging you to complete and build up your profile as you master these basic skills. Now we learned how to load data, how to transform data, how to analyze data, and also how to explore different types of outputs, including visual and textual. So if we look at this text, we can now see this is a table that shows us the position of the samples in the principal component space. But it's not so easy to understand. What does that actually mean? So here we have the example of that output. What are these principal components? Why are we getting a 2D scatter plot instead of a 3D scatter plot? So how do we actually understand what this information can provide us with? If you plot the PCA object with a bar plot, you will also get an understanding of how this PCA result characterizes the full data set. And we can see here that the principal component 1 is the most informative component. So it captures the most variability between different samples. But as those PCs continue to add on and altogether form 100%, we can kind of see the threshold. After PC5, they're non-informative. Of course, to get a scatter plot, we cannot visualize all of the data in all of the components. The maximum we can do is 3. So if we visualize that in 3D, we will actually see the relationship between the components and what they mean, and how twisting and combining different principal components together can help us visualize the differences between the samples. So let's learn about how to do this in the next step by going beyond the basics. So if you open the next step, you will see that we will start improving on this basic visualization and try to apply clustering in this multidimensional space to understand what new information we can get out of this analysis. So here you can see that we have a basic way to perform PCA, which is PR comp, and create an object. We can change the way that we analyze. Instead of PR comp using k-means, and for k-means, we all know that we need the number for the k, so the number of expected classes or the clusters that we expect here. Now, if we print this, we will see that an additional level of information is added to our output. However, the output looks like the cluster number 1 combines normal-like and basal samples. So these are 2 different subtypes of cancer, but they are combined into a single cluster. And for example, here we have an overlap between luminal and basal and normal-like. And here we also have 2 different clusters for the luminal. So something doesn't seem right. If we use a different method, so instead of k-means, there's an additional method. There are additional methods called Clara, Fanny, or PAM, which stands for clustering large applications clustering, or for PAM, it's partitioning around medioids. And we can try how these different additional types of clustering can help us understand what is in our data. So if we change from that function to other methods of clustering, we will get a different result. So let's try to evaluate what we are getting in this output. It looks like with this output, normal-like and basal are almost perfectly separated, except for a small overlap right here with basal being in the normal-like. But basal from luminal and from cloud and low are actually separated quite well. We can also see that principal component has a percentage, and PC1 indeed explains 55% of the variance, as opposed to PC2 only 18.34. So let's see if we understood how to write this code. First of all, we have the library already loaded. Can I just run this? Unfortunately, there's no input file either. So now I have to add more of the code. So in this ability to learn about the code, go through the problem statement, and start developing coding skills, what we do is we go through these simple explanations, show how easy it is to modify the settings, and then put a challenge in front of you. Using this challenge, students are going to be motivated because they understand how easy it is, but at the same time, they really have to understand that they're able to do this independently. Each time that a student completes a code block, they are rewarded with points, and those points are being reported under their profile. Anyone that wants to then take this code over to them, so let's say you wrote an elaborate code and you made this code look exactly like you want this, and you want to save it for future reference, how do you actually get the code? Well, it's very easy. At the bottom of each one of your code blocks, you have this copy button, and it copies to the clipboard. And now you can just open this in your RStudio and run it from there. So what this does is it allows anyone to try to work through the problem in the browser, helping understand what is the correct answer, what are the mistakes, and running it right within the browser to not have to deal with any installation or any kind of technical setting that they have on their computer. After they complete this, and you can see that this is the challenge. So after they complete the challenge, they get an additional number of points for completing the whole lesson. The progress is being tracked. So if I go back to my courses, you can see that for those courses that I have started, this is being tracked. And importantly, this is going to be in the achievements right here. So maybe some of you are recognizing some of your names because you are completing some of the code blocks and you're trying to do some of this analysis together with me. So all of this is being tracked. And what we hope to do by offering you a short survey at the end of each course is to understand how the participants learn and are able to apply different methods to different types of data sets, and eventually start offering a recommendation. So really to make it personalized and offer the suggestion for what you should do next based on your objective. Now, importantly, all of this information is linked in with your account. That account is linked also to our main educational portal. So if I go here to the educational portal, for those of you that already have an account, I can also see that you're completing those steps, and I know what we are working on. So all of these people are working on principal component analysis. All of this information is stored under your profile, combined with the theoretical courses that you take on the educational portal, as well as the pipelines that you run on the server, that tBioInfo platform. Not only that, the tBioInfo platform is also connected by offering short scripts for different types of methods that you can directly put into the code OmicsLogic playground to try and rewrite the scripts that the platform uses as you do different types of analyses. For example, if I go to my pipelines, I know that recently I ran a PCA on the platform. Inside the outputs, there is a result file, but I can also get an R script if I was using not the regular PCA, but the PCA R library. So from a perspective of an educator, I can build the experience to go from very simple and theoretical to practical in the sense of a user of bioinformatics to a person who's familiar with what stands behind each button and how they can develop independent skills to continue and develop their bioinformatics capacity. I hope that this was an informative demonstration. Again, we have options for both R and Python. And if you have any questions, please put them in the chat. So I'll be happy to respond to them right now before I move on to how this looks from someone who wants to contribute to this platform and develop tutorials in Python and R using the Code OmicsLogic playground. Okay, so not everyone at once, I hope, and keep your questions. If you have any other questions, you're welcome to email us to marketing@pine.bio. We'll be happy to continue to speak with you about it. So the courses that we've developed so far cover a basic introduction to bioinformatics, developing a sense of data science, and some basic courses in genomics, metagenomics, and transcriptomics. Okay, so there's a question. If I want to load an existing file on my desktop to the platform, how am I going to do that? So I'm actually going to show you in a second what the backend looks like and explain how you can create your own tutorial right here on the platform, storing your data and images within the course. So here I have, and by the way, for if you mean as a user, as a student, how do you load an existing file on your desktop to the platform? I can show you that in Python in just a second. So the objective of the courses that we have is to go from the very beginning by loading files to then knowing how to visualize the data and doing summary statistics, analyzing the data for variance and other types of statistical characterization, doing exploratory analysis, and eventually being familiar with the different types of challenges addressed in genomics, metagenomics, transcriptomics, and other type of data. As you know, these courses are project driven. So we made it so that people can try to apply the same methods to their own data without having to rework everything, but really understanding the logic and being able to simply replace the files with whatever they are interested to use. Now, I showed you today how it looks in Python, in R, and I want to show you in a couple of minutes of what it looks like in Python as well. So let's go back and load a different course. So I'm just going to go back to the courses page. And we'll go to getting started with bioinformatics in Python. And I'm going to load this course called Reading DNA Using Python. So this is a part of their Introduction to Bioinformatics course, and it's fairly simple. And it's going to be quick, I think, for you to understand of what it does at the basic level. And then I'll show you a little bit more advanced course that you can load files from your computer as well. Okay, so once I'm here, you can see that the explanation goes into the actual code of DNA. So we have to deal here with sequences. So this is, the type of analysis that you would want to do, and you would want to maybe align sequences and compare their length. And what you read here in the beginning is how the code actually works, what it does. And at the end, you've learned the code that will create a program to analyze DNA. So then you can see how here we have a simple code to analyze a certain sequence. So for example, try to print the length of a DNA sequence 10 bases in length to complete this task. So following the instructions right here, if I have a sequence, I can measure how long is that sequence, right? So here, if I have my first piece of code, I can paste it right here, and let's try to run it. And I have success running code, and the answer is correct. Now you can also see that I can drop a file right here. So I can literally take any file from my computer and just drop it right here for this browser-based analysis to recognize that I have a file stored in the browser cache. And I recommend that you go through some of the other tutorials that we have to see how you can use that option to try to analyze different types of data, producing visualization, statistical analysis, and even training and evaluating machine learning models. For those that want to use this in R, it's not exactly as simple by just dragging and dropping the file here. You do have to upload your Excel file to some kind of a cloud system. And so for those of you that are just users, it's easy to leverage GitHub or maybe Google Drive to try and do that. But if you are a developer and maybe a faculty member and you would like to develop additional resources for your students to learn coding in Python and R, I want to show you how easy it is to create such tutorials that you can publish on the platform. And by the way, if you are interested, go ahead and reach out to us and we'll be happy to set you up with an account so you can try this on your own. So you can see here the backend of this platform. It looks exactly the same. I see all of the courses that I created or the courses that other people have created. So these are my courses. And I can add a course. So when I add the course, I need to add description, maybe an image, something like that. And I already have it set up. I have it set up right here for this test course. It's not published and not released, so no one can really see what I'm doing. Let's go in this course. Here I added a couple of lessons. and a test lesson that we can try to modify, just so that you can see how easy it is to do this. So in the back end, I have a few options. I can make this into an R code or a Python code, so you can determine what it's going to be. Here I can choose a file if I need to, a description, and save, and I can make this a free or a paid course. And here I can add steps. So if I have multiple steps, for example, in the beginning, I would like to maybe just explain what this is going to be about. Once the people understand what this is about, I want to reward them. So I want to give them some achievement points right here. And then I can add either a text block. For example, this is a test course. So I can actually write this here and I can modify the way it looks. Or I can even add here just directly by copying and pasting from a course that I already have. For example, if I have a course here on phylogenetic analysis, I can just take all of this text, including the images, just copy it and paste it over to see it pop up exactly the way it was. So it's really simple to go from existing drafts that you might have to a course that is going to become interactive. How do I actually add that interactive portion? Here, I can add videos, images, code. So if I want to write some syntax, so library ape, for example, right? And I can also add different types of code blocks. So for example, if I created the second step, try this out, and I'll say code tried. And then here, I'll give it 200 points, for example. And I want to add a code block. So here in this code block, I can just take this code that I already have written out right here, write it in here, and test it on my computer to know what is the expected answer. So I just place the answer that I expect to get from this analysis and give it a number of completion points to know how to evaluate the result. So as I go through this process, I can create a step-by-step tutorial that introduces the code block, or maybe even the specific data set that I want to explain, the challenge of that data set, then help by understanding the syntax and trying to run that syntax on your own, and maybe creating a challenge with a specific outcome that could be the exact same output that I'm getting in my RStudio or Python developer environment on my computer. So it's really simple to add this to the repository, to the library of different resources. For those of you that are faculty and are interested in contributing, please do reach out to us so we can show you how to create an account as a faculty or as a student. The courses that we mentioned, again, if we go through some of the courses that we have right here, are going to be expanding. Every week, we hope to add some new resources. And in fact, the course that I was just showing you right now is added right here to compare DNA sequences using phylogenetic analysis. So we have a full description of the ape library where you can build phylogenetic trees and analyze similarities between sequences. And we will continue to expand this to other areas as well. Now, before we complete the session, I also understand that there's a big demand for those of you that might be interested in something like this, but don't know how to get started. So let me briefly introduce how we provide hands-on training for this kind of coding bioinformatics, linking together the different elements, the theoretical, the practical, and the skill-based, like coding, into different types of programs. For those participants that are interested in learning and developing a project of their own, we have a program for advanced users. And for those that are just getting started, we have very introductory programs that require minimal technical background and will introduce them to the different ranges of applications of bioinformatics. So a number of different bioinformatics projects. For example, right now, we are running a program called Introduction to Bioinformatics. This program is designed to provide the very basics without any technical capability, illustrating how the different processing, analysis, visualization, and interpretation methods can help you understand the application of bioinformatics to cancer biology, agriculture, neurodegenerative diseases, and infectious diseases. So in this program, we have 6 different sessions that are interactive. So we meet twice a week. We discuss different theoretical aspects that are supplemented by independent activities that they can complete on the educational portal, on the server big data analysis platform, and on the code playground. Now, the code playground is optional for the introductory course, but it is something that we go in much deeper as we continue with the data science program. So the data science program is actually designed with a much greater focus on the application of different types of analysis methods to understand different types of omics data, understand what happens behind the scenes of each one of the buttons, and also is an excellent introduction into the domain of machine learning for data mining and for predictive analysis. So we put an emphasis on such things as different methods of clustering, application to different types of data sets, and classification with different models, model evaluation, and eventually a number of projects that you can develop so that you are independent in what you do. Okay, so the question there is the data science program part of the premium code package, or is there another fee? These interactive programs are comprised of different aspects of these elements that we presented today. So Bipsa, thank you for pasting that link. I will just briefly show you how you can learn about the pricing of the different elements. So if we go to the data science program, I'll just move this out of the way. Data science. The data science program includes access to programs, to courses, to the code playground, interactive sessions, and also independent projects that you can develop with our tutorials. You also have some advanced options as well if you want to get mentor support, or if you have a project, a research project in mind, you can actually add on educational cloud pipelines. So you can run pipelines on our big data server, because that's a major limitation for those that want to work with big data sets from cancer biology and others, that really gigabytes and terabytes of storage are needed to be able to process those. So there are a number of different options for this. These programs start and end at a certain time. So the data science and introduction to bioinformatics already started this week. If any one of you wants to join, we only did introductory sessions up until now, so the registration is still open until the end of this week. But we will continue to repeat these programs every year. So the programs are built around a 6-month cycle. And we also offer a research internship. And the research internship is really designed for those that want a more flexible schedule. And they really prefer to work on independent projects instead of following a specific curricula. So if we are just following the existing curricula that we have with dates, with set up times and topics that are run, you join a training program. If you want a flexible schedule and you want to work on an independent project, we actually have a number of students that are working on their own research projects, and we have a number of examples of what those projects look like, if you would like to know. And those are also available as a part of a regular interaction with a mentor, and you can actually find this fellowship information, we call it a research fellowship, right here on this link. Okay, just a couple of questions, let me address those. Okay, so If a course has a star, that means it's a premium course. If you have a subscription, a basic subscription, I'll pass it on to Mohit here in just a couple of minutes to talk about the basic subscription. We offer a very basic beginner subscription for code, OmicsLogic.com, or for the educational portal that includes everything you need to do asynchronously at just $10 a month. So you could really do this at any time. And yes, so most of what we talk about can be done in the cloud, and that's what it's built around. But if you want to do it locally, you can obviously do that as well. You just don't get the points that we keep tracking in our system. Okay, so let me pass it on to Mohit Mazumdar, who will explain how the basic membership works and what types of resources are included in the basic membership for those of you that are interested in the asynchronous options. Mohit?
Thank you, Ilya. Okay, hello everyone. I'm Mohit Mazumdar and I'm one of the global representatives of Fine Biotech. And in this role, I'm helping students and researchers to learn about big data in biology. Today, I want to take you through our online courses that are now available for everyone to take bundled under the basic subscription. So if you want to learn about transcriptomics data analysis, here is a complete guide of RNA-seq analysis. Transcriptomics 1 is an introduction to next generation sequencing to study the RNA-seq expression data. Transcriptomics 2 is a course about gene expression analysis and focuses on studying variations in expression between conditions. So you will learn about factor regression analysis and t-test and learn about the statistics applied for this big data. Transcriptomics 3 is a course dedicated to analysis, advanced analysis methods, such as machine learning algorithms that allows us to find meaningful patterns in big data sets, especially complex patterns that are typical in these RNA-seq experiments. And Transcriptomics 4 is a review of single-cell RNA-seq data analysis and a study of time course data for thousands of single-cell samples. So if you're interested in transcriptomics data analysis, I would recommend you to check this course series. And if you're interested in genomics, if you're interested in learning about DNA and changes that happens to the genetic code, the genomics series, of course, explains how next generation sequencing can be used to study genome-wide genetic variation that results in phenotypic changes. So the introduction to genomics is an introductory course, introductory and a free course. And Genomic 1 covers practical application of genomics to research and medicine. So you will have pipelines that you can build in these courses. So that gives you a hands-on experience. So Genomic 2 focuses on the analysis of single nucleotide variations, SNVs, and genome-wide association studies, GWAS. And if you're interested in diving into the world of microbiome, then please review this 3-course series about metagenomics. So Introduction to Metagenomics is again a free course, and it will familiarize you with the ways microbiome influence their hosts, and also you will learn how you can study this unique community using the NGS technology. So Metagenomics 1, however, focuses on the impact of bacterial diversity on human health. So you will learn about multiple types of sequencing methods such as 16S, RNA, and DNA, and then practical analysis of 16S sequencing data using the TBioInfo platform. So in Metagenomics 2, you will learn about you will learn to compare microbial composition in different conditions, and then learn about 16S amplicon sequencing, and that also covers data2 analysis pipelines. So for advanced research. And the fourth type of data type, the fourth data type is epigenome. And if you're interested to get an introduction to mechanisms of this non-coding gene expression regulation, then I would recommend this epigenetic series, and where you will learn about the regulation of gene expression with regulatory RNAs, DNA methylation, its mechanisms, assays to find it, and study it, and ways to analyze this data. So the data that is associated with this assays, such as bisulfate sequencing, histone modifications in epigenomes, and also chip-seq data analysis. And last, so many methods in bioinformatics, as you know, requires this application. So in-depth understanding of machine learning algorithms and tools. So in the specialized course on machine learning, we developed a series of courses for practical tutorials that provide a solid foundation for students on this important topics that are often misunderstood because of this boom and all. So topics that we review in these courses includes data preparation, data mining, classification, model training, validation, and many others. So in this course series, you will learn about computational innovation and algorithmic improvements that have been quite important for this biomedical data discovery that we are seeing. So as Ilya told you, all these courses that we discussed are now bundled under a single basic subscription, and which is available only for $10 a month, a monthly subscription. And to subscribe and start your bioinformatics and GS data research, please use this link. Picha, if you could share the link for registration for the basic subscription. And with this information, I will pass it on to Ilya to take this forward. Thank you, everyone.
Thank you, Mohit. And thank you, everyone, for joining us today. I did want to mention that we have a number of options to get started for free. So I do hope that most of you that are here today with us will be able to get started for free, give us feedback, and try out some of these resources, both on the EDU portal. You can try for free. On the Code OmicsLogic Playground, you can also get started for free. We're very interested in your feedback, so please do let us know what you might have in mind for how you can use this in your either education, in your studies, or in your training as a faculty member. And we do have our team here with us, which includes support at PyBio for any questions, any technical questions that you might find on the platform in the coding or the courses, and as well as any kind of marketing questions. If you have any questions about registration, any question about the different programs, you can contact marketing at pi.bio. So Deepsa, if you could please put in that contact information here, thank you. And if you're interested in developing resources for your own training, I will be happy to interact with you personally. My email is ilya@pine.bio, and I'm looking forward to discussing the options for that. Now, those of you that would like to receive a recording of this webinar and see some of the other methods and resources that we have, please visit our YouTube channel. Please, Bipsa, if you could paste the link to our YouTube channel as well. And obviously, we will follow up by email to share with you the recording of this webinar and some instructions on how to get started that we covered in this session. Thank you again for joining, and looking forward to continuing to see more engagement in these different courses that we discussed today. And we will have another webinar very soon to continue and talk about how you can build programs based on some of these resources that we have discussed.